Okay, welcome to those of you who are joining us today for our Monday Multiomics seminar series. Our first speaker today is James Cox, who comes to us from the University of Utah. James is the director of the Metabolomics, Proteomics, and Mass Spectrometry cores, and the director of the cores here at the University of Utah. James' expertise is in mass spectrometry, and today he's going to talk to us about metabolomics and the important use of metabolomics in analyzing your cell types, your tissues, and hopefully helping you in the field of hematology to understand good metabolism studies. And James, thank you for talking today. James has agreed you can interrupt him if you have a question. And I just want to let people know this is recorded and we post these talks, these lectures on our YouTube channel. So if you missed part of it or you missed one, you can go to our CCEH website and find the YouTube channel where you can go and listen to this talk or talks in other series that we have provided over the years. And we'll turn the time over to James right now. Thanks, James.
Great. Thank you, Diane. So today's talk is a little bit different from my usual talks. This is not as technical as I usually do, mainly because the user group or the audience is not probably super interested in the fine details of mass spectrometry. So I envision what I'm going to be talking about today is more useful for the user and how really to think about metabolomics experiments and how they will help you in your research and actually how to work with us as a core as well. And so I'm just a couple of slides of a brief overview of like who we are and what we do. And so who we are, we were started 20 years ago by a Center of Excellence in Molecular Hematology grant to Jerry Kaplan and Jim Kushner, and they hired me. And we started off with one mass spec, the metabolomics core, and now we have 10 mass specs over here. I kind of showed this. We're doing all kinds of different things. We have GC mass specs, LC mass specs, everything you can imagine, and probably not enough. But also, we have a number of really good people we've hired over the past few years. Alan Mashik's the associate director, and he does lipidomics. It's something we're really strong at, and he'll be giving a talk in a month or two. We have recently hired Paul Stewart. He's faculty now in the Nutrition Integrative Physiology Department. He's the associate director for doing informatics for us, and has really brought some of our informatics up to a much higher level. He's only been here a month, so he's doing a pretty good job there. We have Leon Cotreau and Quentin doing metabolomics, and we have some research specialists doing sample prep, etc. And we also added another person who should start next week, and we could probably use a couple more. Over the past 20 years, we've been pretty productive. I've got a couple hundred publications out of the cores, and these are just the sort of the last few that we've done with Diane's on these, a couple of these, I think. We just had an exciting cell metabolism paper accepted last week with the Galmazzi lab at University of Wisconsin with heme and brown adipocytes, and a Nature Metabolism paper accepted as well last week at the Katsu Funai lab. And so what do? We do non-targeted metabolomics, targeted metabolomics, isotope tracer analysis, non-targeted lipidomics, targeted lipidomics, and absolute metabolite drug quantitation. So really these are all things mass spec that relate to small molecules that are found in the body. But today I'm really going to only focus on two aspects. That's the non-targeted metabolomics and the targeted metabolomics. As I mentioned earlier, Alan's going to talk about the non-targeted lipidomics and the targeted lipidomics in a couple months. We do a lot of isotope tracer analysis. I'm not going to really talk about that. That's kind of its own special field. And absolute drug and metabolite quantitation. It's pretty expensive to do, and we do it, but it's not something I'm going to really discuss since we don't do a lot of it. And so, just we're all on the same page, just a definition of what metabolomics is. It's the large-scale study of small molecules, commonly known as metabolites. I've just pulled this off Wikipedia, so there's a dozen other definitions that all sound the same for what this is. But all these metabolites are found within a cell, body, tissue, organism, and so we call how these molecules interact as the metabolome, or all these molecules, how they interact as part of the metabolome. And what I really want to stress right now is no single method is available that is capable of fully annotating the metabolome. We're just going to kind of go through this point in some finer detail in the next 40 minutes or so. And so, where does metabolomics fit into this omics pipeline? So, that we're going to be talking about in the next couple of months. Well, metabolomics is and metabolites are at the very end of the pathway. And it starts off with DNA and RNA, as we all know. So, DNA has the code, and these are pretty complex, but at the very basics of it, pretty simple. Um, Molecules. They're very long polymers of four bases in genomics, or very long polymers of four bases transcriptomics that aren't as long for RNA. And these are really easier to get at the complete sequence of these using sequencers, right? And so we can get a whole genome sequence within a couple of days. And with some types of genomics that we do, we can do a whole genome from a parent, a child, and in like 3 days, really quick. RNA is the same thing. We really rapidly process RNA data. And now we're coming to proteomics, where just recently there's been a revolution in this field as well. We're able to sequence, we just did PBMCs like macrophages. We were able to get 7,000 proteins really easily in a 90-minute run. So, and the reason why all these proteins, RNA, and DNA work out really well is one, they've been around a while longer, so the technology is developed much quicker, or actually have more time to develop. But they're all polymers, right? So proteins are 20 amino acid polymers, so we can really develop methods um, for the analysis of DNA, RNA, and proteins based on the fact that they are repeats, very simple molecules. Where things differ is in the metabolites. Metabolites aren't polymers for the most part, and they're all individual molecules. So metabolomics is estimated that between plants and bacteria and humans, there's well over 100,000 metabolites. I'd probably say there's well over a million. In the human body, though, there's probably only a couple thousand, and most eukaryotes probably have less than that. But what makes this unique, metabolites, is they all have different structures, right? So you have fatty acids here, you got a fatty acid chain and acid. You got amino acids that have amino groups, as well as carboxylic acids. You have sugars that have a bunch of alcohols. You have ATP or ADP shown here, and they have different phosphate groups, sugar groups, etc., and lipids, which are also pretty complex but unique compounds. And so what this says is we really have to have alternative strategies for the analysis of the metabolome um, versus the proteome and RNA and DNA-seq. And so I divide the metabolome into two different arbitrary classes, the low molecular weight metabolome. And these are the metabolites that are, well, low molecular weight, and what we usually would see in our biochemistry textbooks, right? So this is a TCA cycle. And the low molecular weight metabolites are, citric acid, um, isocitrate, alpha ketoglutarate shown here, succinate. And these are highly polar charged molecules that are fairly small with a number of stereocenters, right? And we have these larger metabolites, and I just shove NAD or succinyl-CoA shown here. And so that gets into the larger molecular weight metabolome, which I have shown here. And these are just as important to metabolomics and understanding the metabolism of cells, these very large metabolites, as our small molecule metabolites. But because they're so much larger and they have so many different chemical properties, we have to develop alternative strategies to really get at the analysis of these compounds. And so what this brings us to is, again, there's no single method that is fully capable of analyzing the metabolome. And so this brings us back to what I really want to talk about today is how the metabolomics processes and the user and the core can interact effectively. And so I divide this up into three different stages. The first stage is a user-driven stage. The user has a hypothesis, they design a study, and they perform an experiment. And they bring those experiments to the core, where we do the sample extraction, the instrument analysis, and the data extraction. And from that, the next step is for the user to analyze the data and to interpret that data. Now, there is definitely and needs to be a lot of interaction between the core and the user at a number of these stages to really design a really good experiment. But for the most part, this is how I see um, us as a core interacting with our user groups. And so the first step is really hypothesis, right? And so we get a lot of criticism, not so much lately, but early on in my career is, metabolomics is just fishing, proteomics is just fishing, because you didn't have a hypothesis. I would argue that we always had a hypothesis. We always had a hypothesis whenever we did any of these experiments that it's a high level hypothesis, but altered metabolism will be observed between two or more groups if you do some kind of manipulation to one of the groups versus a control. And or otherwise, you wouldn't be spending the time and the money to do metabolomics or proteomics, right? And so, altered metabolism, as part of this hypothesis, it can come from a number of different sources, right? Very common early on, we were doing a lot of genetic manipulation on yeast. We do a lot of work now with diet, um, disease. We've done disease states, we write healthy versus control, drug intervention, environmental factors, different growth stages, etc. And so then the next stage is to take your hypothesis, right? So what are we looking at, a knockout versus control? And how do we next design a study or a strategy to look at these changes in metabolites? And so I have two different stage strategies here. One is a non-targeted metabolomics approach versus a targeted metabolomics approach. The non-targeted metabolomics approach is often we use this for biomarker discovery. And this is just a broader metabolite coverage where you're just looking for anything that changes. And a lot of times we're not even looking for to identify every possible metabolite within the metabolome or detected in the mass spectrometer because it really becomes overwhelming. We're just looking to identify metabolites that are changed under a condition, right? And so, because it's such a broad analysis and you don't have the time or the money to really target each individual metabolite if they don't change, it really is a qualitative approach at metabolomics. And really, it's very good for biomarker discovery or just further hypothesis generation. But most people, when they do science, they have a pretty good idea of what is going to be altered in a pathway wise, right? So if I alter a gene like a conatase in the TCA cycle, I'm going to pretty much predict that there's going to be change in the TCA cycle. So you can use that knowledge of what you're doing in an experiment to design a metabolomics approach that's effective. So the thing about the targeted approach is it has a much more narrow metabolite coverage, and therefore you can do a more semi-quantitative or quantitative approach to analyzing these samples. And when you have a much more a quantitative approach, you get much better statistics. And also you can really,
Could I just ask a quick
yeah,
question?
absolutely.
Thanks. How would that apply to like prostaglandins, eicosanoids, and kind of really, let's say, reactive oxygen species that are, sort of mitochondrial or luminal.
Yeah. So
How do
that's
you,
where you have to ask the core how you're going to do that. That's a really good question because a lot of those studies like that, that's a very ex specific extraction technique because you really want to keep the redox state from not changing, right? And so we have very specific extractions for that. And then we have a very specific targeted quantitative method to measure those using our most sensitive instrument. And that is something we've really started working. We have a method for that, but it's really come online the past 2 months.
well,
So that would be a targeted approach. And that would a lot of these pathways like this, you wouldn't even use a non-targeted approach because the problem with non-targeted is it's not super sensitive.
Great. Yeah, absolutely want to hear more. Thanks.
Sure, no problem. Any other questions? All right. So, um, so the targeted approach is also hypothesis validation. So if you saw something different, and going back to Bruce's question, a lot of times, non-targeted isn't really sensitive. So you might see a change in the most abundant metabolites in that particular pathway, but you don't always observe the other metabolites in it. So you can take your hypothesis is, oh, pathway TCA pathways alter. We only saw changes in citrate. You do a very targeted approach, and then that will the target approach, you hit all the other enzymes in that particular pathway. So then the study design, the further part of the study design is, which mass spectrometry method do you want to use, or which approach is best for your particular question. As I mentioned before, no single extraction or analytical method can capture the entire metabolome. And so what we use is GCMS or LCMS, and they have their disadvantages and advantages, right? Um, we we've been using GCMS really to target the small molecular weight metabolome, like I showed earlier, the TCA cyclointermediates, organic acids, amino acids, and these things like carbohydrates. Like is GCMS is really excellent for that. Whereas LCMS is really good for redox metabolites, the things that are larger, the higher molecular weight metabolome. NADH, NADPH, AMP, ATP, CoAs, and nucleotides. And so the LCMS is actually much more sensitive as well. So we were really trying to find low molecular weight metabolites in a targeted approach, or low abundance metabolites in a targeted approach. We'd use LCMS. But GCMS is really quite an excellent approach because it has a much higher ability to separate um, compounds. So it has something called theoretical plates. There's a number of compounds you can separate in a short period of time. It's much higher in GCMS than in LCMS. And so that allows us to separate carbohydrates very efficiently. So C6H12O6 is the molecular formula for glucose, but it's also a molecular formula for about 30 other carbohydrates. And we're able to, using GCMS, separate all those different carbohydrates out from each other and get an identity for like galactose and a number of those other isomers. And so the way GCMS works, it separates compounds by boiling point and hydrophobicity. And like I said, it has something like a very high peak capacity, so you can separate out isomers from each other very easily. So we can separate out, as an example, isoleucine from leucine, which are isomers, by a retention time of a minute. So there's really, really clear separation between those. But the downside is it takes more steps to do it. You have to use some chemistry to change, in this case, oxalic acid, I think. So you have to convert it to this apolar state, and that takes time and money. And the other problem is GCMS has an upper molecular weight limit. And so that's where I really start separating the low molecular weight metabolome from the high molecular weight metabolome is its ability to be processed by GCMS.
I don't want to keep slowing you down, but
Sure, no, go ahead.
I thank you so much for like letting me hear the phrase theoretical plates. It's been probably 40 years since I've ever had that. And it used to be something so close. Anyway, but I was just thinking of with like ion trap or something like that, where instead of doing the pre-sorting, could you do like a voltage dependent like whatever you call it,
Yeah,
fragmentation of the molecule that then tells you something about the stereochemistry and the like structure, even if it is at the same molecular weight.
we do that occasionally, but the problem is when you start fragmenting like that, you start losing sensitivity. So like an ion trap where you're going from MS1, MS2 to MS3 and to separate those things, that's certainly a possibility. But you really start losing the sensitivity by like 1,000-fold as soon as you start MS and
Wow.
MS.
Okay.
And then the other problem and this is changing a bit, and I think this is more technical, but as we move from the DDA approach, this is getting really technical, to a data independent analysis, that will happen more often, but we're not there yet for the metabolomic side. And that's happened on the proteomic side, and that's when there's been a big revolution in proteomics in the past year. Is those are coming, but not really from metabolomics yet.
And then
Um,
stuff like absolute GSH versus like relative reduced to oxidized glutathione profiling, is that um, pretty robust, or is that
Yeah, so we have an approach for that. So what we do um, is we take a sample and we dramatize it under the conditions where it's not going to go from GSH to and dimerize to the GSSG. And so what we do for that, we extract it and then we immediately derivatize it, the free thiol. And so that immediate derivatization, the free thiol, and we met to prevent the
empty?
cross cysteine bonds, allow us to get the GSH to GSSH ratios from that. We'll actually add in an internal standard for that as well if we really, really wanted to get absolute quantitation. But we can use that internal standard also to see how effective our derivatization technique was.
Fantastic.
Yeah, so LC-MS metabolomics, something we do a lot of as well. This really has some really strong advantages. It doesn't require derivatization. It's super sensitive. Like if you really want to go after something that in low cell number or low abundance in cells, is you're going to use LCMS. The problem is it's difficult to separate compounds completely from each other because the liquid chromatography just does not have the number of theoretical plates compared to GCMS has. So that's the compromise. A lot of projects will do GCMS and LCMS on at the same time.
I wonder if there's a like a metabolomic signature sequelae biomarkers of having had low GSH levels for some period of time. So then you see this like scar of oxidation across a whole bunch of metabolites or
Um,
protein.
that's a good question. I don't know, but where you would see that probably is on proteins, since a lot of those proteins get
Um,
glutathionated. And so if you really wanted to look at that, I would look at like probably albumin and see if there's been a change in the glutathione levels and something like that. But then you have to think about protein turnover. So
yeah, and then also compartment specific markers too that could be more like if there's an organ specific or cell type specific disease process going on,
so we
then
do that for mitochondria for sure. So there are certainly patterns that we observe for dysfunctional mitochondria. Usually it's TCA cycle or intermediates or a shift to fatty acid oxidation. And so that's one of them ones we'll use. And there's a couple others. Yeah. All right. And so then the next step you want to think about is study design, and study design is sample numbers. And this is something that you want to talk to the core before you process your samples. Is really the more complex the sample, the more biological replicates you're going to need. And so my favorite model organism of all time is the fruit fly, the Drosophila. And the reason why is these particular critters are so inbred that you create a genetic mutation in them, you'll see that in a metabolism really readily because the controls have super, super tight CVs. In other words, if you process, 6 sets of flies, the percent difference between those 6 different flies will be minimal if they're all the same strain, right? And cell culture is pretty similar to that. We probably need 6. There's actually cell culture's got more variation that I've observed in the past than fruit flies. Um, if you want to go up a little bit more higher in complexity, I think the mouse is probably way higher in complexity than a fruit fly. But because they're so inbred, like 6 to 10 mice work pretty well. Uh, Same with zebrafish. And then when we get to humans, and that's when things get ridiculous. So it takes thousands of human samples to reach power to determine the differences between populations. And that gets super expensive, right? And so we do have projects where we're analyzing 2, 3,000, 5,000 humans. Um, and those eat up a lot of time and a lot of resources to do that. And the other one thing I want to point out here is metabolites have a wide dynamic range. In other words, the highest concentration metabolite can be 1,000 times higher than the lowest concentration metabolite. But the problem is the mass spec only has a 5 to 40x dynamic range, and that's really dependent upon uh, what you're analyzing and the mass spec itself. And so when we do some of these experiments, we'll actually do different concentrations, especially on the GC side. We'll do like a high concentration analysis so we can get at the low molecular metabolites, and then a dilute um, analysis so we can get at the really high concentration metabolites so we get them out of a saturating range. And so those are
James?
expenses. Yes, ma'am.
Oh, sorry, I'm going to just interrupt. There was one
Sure.
question in the chat.
Yeah.
They're asking, you're referring when you say 4 to 6 or 6 to 10, that's per group. So if you have a
Yep.
wild type and then 6 to 10 mutant mice, right?
Yep.
Yeah.
Hmm.
Does that answer your question, Randy?
Yep. Thank you, Diane.
You're welcome.
So I don't have the chat up. So Diane, anytime you see something, let me know. All right. And so then we get to the experiment, right? And so the key to do really good metabolomics is consistency. And I can't stress this enough. I think this is the only thing I've really highlighted in any of my slides is consistency really is the key. So when you're planning an experiment, plan to minimize any batch effects. Because we'll be able to tell the difference between in almost anything. Like if you collect samples on one day and you wait a couple of days and you collect your mutant a couple of days later, we'll be able to that's probably be one of the first things that we're going to be able to tell if we do like a PCA analysis is that, yeah, you got batch effects. So it's super important to collect all your samples on the same day if possible. And I realize that's not always possible, but really try to do that, especially on simple experiments to collect. Um, use the same pipettes, the solvent standards, use the same technician to do it. Just be as consistent as possible to minimize those effects. Other examples of consistency is same lobe of liver. We do and have done a lot of liver metabolomics, and the liver's got a large amount of variability across it, and so we prefer really to get the same lobe of liver um, from every sample. And if you can, and Judy Simcox, when she was here at the U, she's at Wisconsin now, she would freeze clamp the the livers and the mouse, and then use very specific lobes to do her work, and that worked out really well. She had outstanding data. Other example is cell culture. Make sure they're all the same confluency, um, same amount of plasma. So um, people have given us like wide-ranging number amounts of plasma, and that's very difficult to sometimes we don't know if it is we have like 25 microliters of plasma in one tube and 35 in the other. We don't know that. We're not told that. Then we'll extract everything the same. And actually, that really distorts the data as well. Same number of yeast and bacteria cells and same amount of tissue. And make sure you weigh that tissue before you give it to us. And what we really want with all this is really having a highly curated metadata. So make sure you talk to us first before you drop these samples off, and we should discuss all these key aspects of the experiment. But we really need to know the exact amount of plasma you have given us and what's in the tubes or the number of bacterial cells, etc. And so this is key, really the biggest key for having a successful experiment is consistency and high curated metadata. And really everything else kind of takes care of itself if it's a well thought out experiment. So the next step, and go into a little bit more detail because we do it, but we also have a lot of labs do it on their own. It makes things faster overall as sample extraction. So the overall goal of sample extraction is really to minimize metabolic alterations at the time of sampling while maximizing the metabolite recovery, right? So the idea is you want to freeze metabolism at the time of sampling using a freeze clamp, like I said, Judy would do or others have done for tissues. Um, really cold solvents. And then what you're doing tissue, you're rapidly disrupting these in cells and tissue using bead rupture tubes, right? But by far, regardless of any approach you're doing here, and please contact us in the core. We've done every extraction possible known to man, but consistency is the key, right? So if you're going to do it one way, always keep on doing it that same way every time you do it. So I'll go through this really quick. Traditional work, sample extraction for plasma. It's pretty easy. It's just solvent extraction. So we just take 50 microliters of plasma and we add a cold organic solvent, usually acetonitrile and some internal standards. Keep it at -20 for an hour, spin it down, and for GCMS, we'll dry it down, or LCMS, a lot of times we'll just run it right on the mass spec. Um, we will train people at the U or other people. We've actually have a postdoc exchange where we've trained people on how to do these experiments in their lab. And that way, if you want GCMS analysis, you can just dry these samples down and you don't have to send them to us on dry ice. You can just send them to us in a box, well labeled sample tubes, and we can run that by GCMS. The great thing about a lot of these endpoint metabolites, especially the ones found in GCMS, is they're really stable. Amino acids stick around for hundreds of years for a reason. There's a reason why they're found at the end point of metabolism. It's usually because they're a very stable compounds, right? Um, but the problem with this approach for LCMS, it doesn't remove phospholipids, and this can cause considerable issues with ion suppression at mass spec. And companies and CROs and academics have spent, thousands and thousands of hours and millions of dollars really to figure out best ways to limit the ion suppression effects of phospholipids. And so, and the reason why that is phospholipids will often interfere with your accurate analysis of the metabolome, or if you're drugging humans, the accurate quantitation of drugs in plasma. And so there's two different ways to do this and that we use in the core. One is general ways, liquid extraction. And the nice thing about that is you have plasma sample, you have methanol water and chloroform, and it pellets out the protein, and then the aqueous layer goes to metabolomics. You can do GCMS or LCMS with that. Bottom layer, you can do lipidomics. So that's a pretty convenient if you really want to get at both. The problem with this liquid approach is it's noisy. So we don't get really good statistics or percent CV doing this approach. So another approach that we do is solid phase extraction. And in the CRO world for drugs, lots of companies do this. Solid phase extraction is probably used far more than in liquid extraction. It's because it's got a much higher level of repeatability. And so we use something called EMR Captiva plates. And what that does is that absorbs lipids onto the matrix in these tubes and elutes the metabolites. So those eluted metabolites can go to metabolomics. And then we push out or remove the stuck lipids using a solvent. And we take those and do lipidomics with those. So the next thing I want to talk about before we go further is sample extraction and quality controls. So if you're going to do this in your lab, and we do this quite often, is we're testing samples with a really high level of quality control. And so we have a number of levels of quality control here. And so one is a system suitability test, which is 13C labeled amino acids that we've made in the lab, and we store at -80. And we take those out, and between every batch, we actually will run that to make sure the instrument is performing well and there's no drift in the LC or accurate mass of the instrument. We also do pool QCs. So this is where we take your sample and we take a small aliquot at every one of the samples we extracted, and then we build those into the profile. And so we test each one, every pool QCs, every couple samples. And shown here, one way to do this, we can pull the QCs, the wild type, and we can pull QCs, the knockout, if we're looking at knockouts. But usually we're just pulling them all together as one. And we use these also to follow drift of instruments. So we have multiple layers of quality control. We'll also have blanks. So these are extraction or process blanks. They contain internal standard, but everything but like a plasma or the liver or whatever you're looking at. And so we use this to look for background chemical contaminants in our extraction process. And we also have double blanks, which only contain like the buffer we're running in. And that way we can determine, what chemical contaminants are coming from the system itself outside the extraction process. And so we ask labs if they're going to do the extractions, that they follow this routine as well. And so that way, when we use our software to look at how well the extractions went or how good the data is, this is an important aspect of that, which I'll go through in a minute. So this is just the overall instrument analysis. You're going just, from a patient to sample to these EMR plates. These are the solid phase extraction plates that I was talking about. We give lipids and metabolites in two different plates. And then we can do C18 chromatography by UPLC. And this is what lipidomics looks like. And Alan will talk about this, like I said, in a month or two. And this is UPLC. This is what a UPLC trace for metabolomics looks like. And we'll extract that in data extraction. And so, of course, the core does this part and the data extraction. And I'll show you that next part next, actually, unless there's any questions. Okay.
Just,
Oh,
just
sure.
on that scale, is the eicosanoids are just totally not even in range, or like,
Oh no, a custom. Yeah, yeah. So that's certainly a very targeted approach. So I don't know if Alan's on the.
Yeah, I'm here.
How many microliters are we doing for icosanoids and plasma?
20 microliters will suffice to get icosanoids. It just takes a second. That needs to be a separate aliquot from your global lipidomics run.
well,
Yeah. So Alan's my expert on that for sure. He's the one who developed that assay in the lab. So if you want to contact him, I can put you in touch with him as well. Um,
yes, absolutely. Yeah.
Perfect. Yeah, so the next step is the data extraction, and this can be by far the most time-consuming part of this whole analysis is this section. And so what we're doing here is after we've run and this is I think lipids here, but once we've run the analysis on the instrument, what you see is that blob, right? So you have a blob of peaks here, a blob of peaks here. And what we want to do is we want to extract all those individual peaks into something meaningful. And so the way that's done is we're looking at the accurate mass and retention time of compounds that we know exist. And so in this case, in red, this is acetylcarnitine, and it's got like a mass like 177.8, I think, or something like that. And we know it elutes like 5 minutes. And so we determine the area under the curve for this particular metabolite, which is the red, and we assign that a number, or the software assigns that a number in this particular software. This one's called MH Quant or Mass Hunter Quant. And there's other quant softwares out there. It's all vendor dependent. And so what this software does is it takes all your controls and your wild types and your mutants and finds the area under the curve for all the metabolites that we know of in each chromatogram. And after much staring at all these chromatograms and making sure that the peaks are correct, it'll pop out a data file. But the reason why this is really quite time consuming is our core really looks at all the peaks almost individually in some cases. And so we want to make sure that the metabolite that we're calling to be acetylcholine is actually acetylcholine and not something else. So we do accurate mass, we do MS, and we do retention time matching to determine the identity of all these compounds to be sure what we're looking at is what we're actually calling it. And the end user can be highly assured of our data quality that we put out. So this is the next step here is this is what it looks like after that data extraction. And it's not super exciting, right? But it's a .csv file or Excel file, and it has your sample names and what it is, wild type, and the area under the curve and this is just metabolites that we have in the A's. But really, that doesn't have, a ton of meaning at this point until you do some statistics on it or some further analysis. And so we we next have this program. We call it the Reporterator. And basically, what we did, I had a postdoc, Brad Naylor, do this is um, we put together a little software package that is able to take that .csv file and automatically process it to an endpoint. And sometimes it could be super quick, like a matter of a couple of minutes. Um, and to be clear, we didn't really make up all the R code or the Python code that went into this. We took it from GitHub and another website called Metabolomics, which I'll show you here in a second. And so what we do is that .csv file, we then put in a reporterator and we normalize to internal standards. And then we look at QC of all the metabolites. So what we're doing here is we're looking at all those QC samples that we have and run, and we're looking for any kind of variation or a percent CV that is over usually 30%. And so if our QC metabolites are over 30%, we'll drop those from further analysis. And that way, we're not getting a lot of spurious results. We'll keep those in an Excel file if you wanted to go look at those in the future, but we'll drop those out at this period of time from downstream analysis. And then we normalize to the sum of the signal. A lot of times we'll transform and scale. This is all automated. And then we'll perform visual analysis and we'll generate a report. And so what we'll give you is here on the right is a report, and it can run into 20 to 60 pages or less, and just depending on how many samples you gave us and what we want to compare. But what it does is it tells you exactly what we've done experimental wise and data analysis wise. And the intention here is for you guys to cut and paste that into a publication. This usually is publication ready. So the next thing I want to talk about real quick is where did we get that R code? And we got a lot of that straight out of Metabolomics. So Metabolomics is a website put together by the Wishart Group originally of University of Calgary. And what it is in a sixth form, but it is a way to really go after lots of different data analysis tools by the end user. So now we're getting to the point where, we want you as the user to be able to manipulate your own data and build your own charts, right, or your own graphs. And so this particular program will do that. And the nice thing about the report that we send, we give you instructions on how to do that online. So you'll not only have the data that we sent with a lot of different graphs and charts, depending on what you want, but you can go and see if you can match that up with this particular software platform. And it's completely free. And, in the metabolomics literature, I would say about this period of time, about half the people in publications are actually using it. And so the visual data
It's
yeah.
incredibly well cited software, and it's got so much capability. But I guess I haven't. I would guess that an important part of this is contextualizing normalizations to something of interest, so that you use a group of molecules or something that you might get a little bit more robust normalization about some other molecules being differential between your sample groups. But I'm just totally imagining that and not. I have no idea.
Yeah. So the normalization, there's a dozen different ways to normalize it. Sometimes just internal standard is enough. When you have really large data sets, I like to normalize to sum, and that's the sum area under the curve of all the detected metabolites that you're going to analyze. Um, for really, really large data sets, we normalize to the tick. And that's something like if you have like a lot of molecular features that you're trying to look at, I think that's a good way to go. Um, yeah, so it really depends what you're doing. You don't really want to normalize to sum if you're only looking at, four metabolites because that's really going to distort your data. Usually we just would sum that the lower number of metabolites we have, we would want to normalize to an internal standard. I guess that's the best.
I'd always be looking for, kind of a cross data set approach that could let us see important patterns that in a and have a discovery resource that people could look up and put in some signature that seems to be differential and then relate that to other pathways and things. And some of that's built into Metabonics, I know, but um, um, assembling larger data sets from like data, and how much could we grab all of your data sets and start to string those together across different studies and sample types and and really start to see subpatterns that we think might relate to cell type and
Yeah, the
species?
best way to do that would be a metabolomics workbench, because that's got a ready amount of data already there that's really well annotated. The problem we would have is one, we'd have to ask the PIs, can, can we share the data? And two, we'd have to put all that together, which would just be an incredible amount of time. But if you wanted to take that approach, which is a really cool idea, um, metaboloanalyst would be where I'd first start off looking at.
I like that idea. Maybe once it's published, we can get permission to have a resource of some sort,
Yeah, I mean that's the whole idea of metabolomics workbench.
right?
Yeah,
Okay.
yeah, no, that already exists. NIH has that in place. It's just really it's onerous to put data in. We do it. We're doing it for someone right now, and um, it's not trivial.
I could
That's
just imagine though a lot of study specific batch effects or what you guys have tuned on or other things in your LC type adjustment to really focus in on things if there's any bias. So like trying to say what the pipeline was that was used and then also how it batch varies in your implementation of it would be
yeah.
part of
I
it.
think there's going to be so much batch effects that it's really going to be even within the same lab running the same assay, on different organisms or cell types is just going to be it would be difficult to do. But I understand what you're trying to get at. It would just incredible amount of work. Yeah. So yeah, going from that, my favorite plots, well, I'll just tell you I got two favorite plots that I like. One's a PCA analysis, and this is probably because I started 20 years ago doing this, and this is what everyone did, and they didn't really do anything else. And my second favorite plot is the volcano plot, and we include usually these two in every report we have. And the volcano plot, for those who don't know, and I'm sure everyone here knows, is looking at the fold change versus a p-value. And so these can always be adjusted, the fold change and p-value, to find significance. But it's a really good way to look and see, like this particular mutant had decreased nucleotides shown here, right? And increased citric acid or other things in this side here. And where mutation this was, I have no idea what it is. I can't remember what we did here, but and then we'll send you a list of, these things that change, a fold change and p-value adjusted that we've gone through. And this is all in our report. And then we go through a confidence level. So, how confident are we of the identity of these metabolites? And this is based on the Metabolomics Society initiative for confidence. So anything with a confidence is when we have accurate mass, MS, retention time. They all match with known data that from known standards or standards we purchased. And they kind of go down. So GC just means that we ran it on the GC, and that would have the same retention time, accurate mass, and fragmentation pattern. And so these are all super good matches. Some aren't as good. So this would be an electronic match at the 3. That means we didn't probably run a standard, but it matches the MSMS and accurate mass. Um, so the next step in data interpretation, just a really easy one, is a classic analysis. And this is what I was doing in graduate school, knocking out genes and bacterial biochemical pathways. Is say you have a pathway or you have metabolite A going to B, metabolite B going to C, metabolite C going to D by enzyme X, Y, and Z, right? And so you would knock out enzyme Y, and what you're always looking for is an increase in precursor metabolite B and a strong decrease in C, right? Also, what you'd be looking for is a smaller increase in A, the precursor to that, and a smaller increase in D. And this is super common. And so when we were doing with the Kaplan Lab and Diane Ward years ago, we were looking at genetic knockouts in yeast, and this signature happened quite often. So it was pretty easy to figure out what was going on in these cells. And the way we'd figure this out, say if I had a pathway or organism I wasn't super familiar with, I would use KEGG pathway database. And so this is really nice. You can pop in your organism, whatever you're looking at. You can enter into your search for, like, I don't know, succinic acid or whatever B is increased in, and it would come through with a bunch of different metabolite pathways. And so you can pretty much find out what enzyme is changed there. And so that's fairly easy to do, but nothing's really that easy anymore. And so there are a couple of software tools that are available. One is Metaboverse, and this came out of the Rudder Lab here at the University of Utah from Jordan Berg, and we're pretty grateful to be part of this endeavor. And what this is a pathway discovery tool, or a way to visualize data through metabolomics, and also overlay proteomics data as well as transcript data. And you can get an idea of what's being affected in your metabolic profiles. It works great on yeast and mammals. Other organisms, probably not so much, because it took a lot of mapping, metabolic mapping, to make that work. And the other data interpretation software that I recommend for metabolomics is MetaboAnalyst 6.0. It's my favorite website for this kind of thing. And they offer pathway analysis tools, which they're okay, enrichment analysis tools, and network analysis tools. And you can all use that. But the point I really want to make about data interpretation is the researcher really knows their system way better than the core does, right? So I do not know your particular enzyme of interest nearly as well as you do. So a lot of this data interpretation has to be done at the PI or a graduate student or postdoc level, and these tools will help in that. So just go over some metabolomics limitations in the last minute or two, and I'll take more questions. Is really, I want to get across is no single method can fully scan the entire metabolome. Metabolomics actually has low metabolite coverage. It can be data sparse. And in other words, like, genomes, we can get the whole genome and figure that out. Transcriptome, proteome, we can get 10,000 proteins now in a single cell type. But metabolite coverage is much more sparse than that. There's a lot of holes in the data set and pathways. It's not necessarily sensitive. Metabolites are often at low concentration, and those low concentration metabolites contribute to a lot of that data sparsity. The instruments that we have now, it's actually some of the ones we actually have now are really, really good, and can do a lot of those low concentration metabolites, but that's always an issue. Mass spec doesn't work on every metabolite. Not all metabolites extract efficiently, so many amount of proteins. That brings us to ATP. And so ATP actually is found bound to a lot of proteins. So if you really want to get at that particular metabolite, you have to use either acid or base to get it off a protein that it's stuck to. And when you do that, not every metabolite's stable. So ATP is another one that's not super stable. If it sits around, your tissue sits around, and this is why we want to do everything cold and fast. ATP will disappear on you. And so NADPH and NADH. And it's not always rapid for doing the full GCMS analysis, and it can be expensive. So with that, I'll take any questions if anyone has. And I'd like to thank Alan and everyone else in my lab for helping. And yeah, thanks.
Terrific, James. Great talk. Feel free to unmute yourself and ask. Randy, it looks like you have another question.
Yeah, James, this is Randy Brudkowitz from Indiana University. So if we wanted to work with you, let's say, and we want to do GCMS on some mouse tissue, what would the cost be per sample?
Yeah, so that's a good question. So if you go to our website, cores.utah.edu or cceh.utah.edu, we have a list of that. It's probably about $65 for the sample extraction and the GCMS analysis. We do charge an hourly rate for the data extraction of about $54 for my technician to kind of go through that. It would probably work out 80 to $100, probably a sample. And it just really depends on what you want to look at. Yeah.
Thank you.
Our website has all that pricing if you scroll down.
Any other questions?
Is there a way to do uh, isotope labeling for time, get velocity and flux in?
Yeah. Oh, go ahead. Sorry. I'll let you finish.
No, that was it. Flux analysis.
Yeah, we do. Um, we do for people at the University of Utah because it's super labor intensive. If you want something simple, we'll do tracer analysis. So with at the University of Wisconsin with Andrea Galmazzi, we did it for heme biosynthesis. So he was looking at what precursors were being used and the rate they were being used in heme, and we've done that with several other groups at Georgia. And it's pretty labor intensive and takes a lot of communication between the investigator and us. But if it's simple, like if you want to look at glucose incorporation and like the TCA cycle, we've done that a lot. We actually have a fairly automated pathway for the extraction of that data. But the math, when you really want to do those flux equations, we don't do that. So that's something where you'd have to find someone else. So we actually have someone at the University of Utah, Greg. He's a faculty member of biochemistry. He does that, but I don't know how much he collaborates outside. Uh,
I actually have a very hematological question. I
yeah,
I don't know if anybody knows this. I bet you somebody does. It's this neonatal jaundice that gets really severe if you have some, I think it's G6PD-H or some deficiency that actually diminishes reduced glutathione. And so that if you have anything that's that I don't know this for a fact, but I think it's something along this line. And so the kids that have a really high that get near icteric levels of neonatal jaundice have this incredibly rapid degradation. The fetal hemoglobin red cells are very unstable because of not having oxidative stress protection, and those kids have a very high rate of getting near icteric levels of neonatal jaundice, and actually I think it's an undiagnosed. I think it's they just UV these kids, and sometimes they get discharged so fast from um, after the nursery that it starts happening at home. And so depending on level of insight of the parents and other things when the kids have, I bet you there's a huge number of these kids that are going, they're really getting hurt by having accelerated fetal hemoglobin red cells degradation. Does anybody actually know this? This is something that I just sort of stumbled on in a, kind of genetic.
Nobody on this call that I know of. Somebody speak up if they do, but you could talk to, do you know Bob Christensen? He's a neonatologist here at Utah who actually treats iron overload, iron deficiencies, and jaundices, and he would be a great contact. Let me see if I can find his email. I can post it in there. Just give me a sec.
Okay, thank you, Don.
You bet. Any other questions?
Can I ask a question? Hi, James.
hey,
It's a very informative talk. Thank you so much. And just a quick question on the sample preparation. I have samples of model organism or cell cultures. So how should we prepare the sample and ship them? So in like a frozen pellets,
yeah,
we are not equipped to extract the
no.
metabolite.
So we're happy to extract them for you. Like a lot of people who have like, hundreds of plasma samples, their lab will do that. But if you just want to pellet those cells, remove the supernatant and just snap freeze it from that, we're happy to take it from there. What we want to do is get rid of as much of that media as possible because that contains, a lot of metabolites
Mhm.
um, that can interfere, especially amino acids. They're usually pretty high in sugar and amino acids.
The extraction procedure would be similar for different types of sample, like intact model organism. Like C. elegans has a thick cuticle. I'm not sure if that would affect the efficiency of the extraction.
Yeah, so what we do there is we use these bead mill tubes with the ceramic beads, and that will usually completely destroy the cuticle. We've done um, we've done those before, lots of hard things with those, and it works pretty well.
Hmm.
Um, yeah, we don't really have too much of a problem. This actually the problem more is the size of the nematode, right? Um, I think.
Yeah, for that we can just send a population of the same genotype. They are pathogenic, so let's say we just want to compare the same genotype on a different treatment condition.
Yeah.
Send like a pellet of 400 worms in one single tube. Yeah.
Yeah, that works. So we would just use a bead homogenizer.
Okay,
Yeah.
thank you.
For that too, it's been a while since we've done nematodes, so it wouldn't be a bad idea, to send us a couple extra to make sure we have enough. So is
Mhm.
400 nematodes going to be enough for the assay you want to do?
Yeah. Also, I will check in the literature. People actually have used this. My impression is that people just use the extraction method you mentioned for the regular metabolome. Or not.
Yeah, and we would use the same like cold acetonitrile water as
Mhm.
well, so that solvent would still be in there to denature the proteins. Um, but yeah, we just use the bead mills to really break open the cuticle.
Got it.
Yeah.
Thank you.
Okay, we are past the hour. If you have other questions, I know that James is ready for conversations through emails, or you can contact him. I posted the core website for Metabolomics here at Utah in the chat so that you can use that if you want to reach out to them. You can also see Randy costs of things in there that are posted. There's also protocols posted for those who are interested in kind of getting a basic protocol. But as James has discussed extensively, contact them and they're set up to help you guys be successful. So James, thank you again for the talk. I want to remind others we have another one next month from Kevin Hicks that will be on what's called MIDAS, which is detecting metabolites that bind to proteins and affect their activities. It's a really interesting technology, and I really highly recommend you join in with us on that next seminar in a month. And thanks everybody for your attendance.
Thanks so much.
You.
Thanks,
Okay.
That was
man.
really, really great. Thank you so much,
Yeah,
James.
Thanks.
James, great talk.
Bye.